In our everyday lives, we are exposed to millions of microbes, including bacteria, viruses, fungi, and protozoa. When these microbes come into contact with us, they can establish themselves and form what's known as our microbiome. The microbiome is found in many places in and on our body, from our skin, hair, and nose, to our mouths and gastrointestinal tract. There's been a tremendous amount of research into the gastrointestinal microbiota and its effects on everything from brain function to inflammation. So diving a little deeper, these microbes are found in large densities within our large and small intestines, and when everything is working well, they play a key role in supporting our health. From helping us break down complex fibers, inactivate toxins, create metabolites, outcompeting pathogens, to influencing the healthy development of our immune systems, they play a key role in helping us live healthy lives. In order to maintain balance with the microbiome in our intestines, our bodies have developed a number of defenses to help keep these microbes from crossing our gut barrier and triggering an acute inflammatory response. Firstly, we have a barrier comprised of tightly packed layers of epithelial cells. These cells arise from stem cells located in the deep pits in our intestinal tissue called crypts. On top of the stem cells present within these crypts, we also have Paneth and goblet cells, which create and secrete additional protective barriers made up of mucus and antimicrobial peptides. All of these systems help protect us from the invasion of microbes into our tissues. Within our tissues, we also have a variety of cells that can help mediate immune responses to signals presented by our microbiota. We have a mix of effector cells. For example, lymphocytes, such as natural killer cells and T cells, can mount an effective response against invaders. Other cells, like plasma cells, secrete a protective molecule called IgA, which helps with the capture of pathogens and their toxins, thus acting as an additional barrier against infection. Other cells may be specialized to respond quickly to threats, such as macrophages, dendritic cells, and neutrophils, which have the unique ability to engulf pathogens and degrade them within organelles called lysosomes. Overall, these barriers and cells help to maintain homeostasis, or balance, within the intestines. The microbes are kept at a distance from the epithelial cells via the mucus produced by goblet and Paneth cells. Further effector functions provided by immune cells help limit the probability of an infection establishing itself within the intestinal tissues.
In order to maintain a state of immune homeostasis in the gut, our intestinal immune system must be able to tolerate commensal bacteria and food antigens while simultaneously having the ability to fight pathogens and clear infections. As such, an anti-inflammatory response is needed in order to create a tolerogenic environment in the gut that prevents chronic inflammation. Dendritic cells located throughout our intestines, such as in Peyer's patches, play a crucial role in the contextual interpretation of intestinal antigens. Microfold cells, or M cells, sample antigen from the lumen and transport it into Peyer's patches where it can be taken up by a dendritic cell. The dendritic cell can then process the antigen and present it on its surface so that it is ready to interact with a naive T cell. The dendritic cell then migrates into the T cell zone of the Peyer's patch where it can present the antigen to a naive T cell. Since the antigen taken up by the dendritic cell was a harmless commensal or food antigen and the surrounding intestinal tissue is healthy and not damaged, the dendritic cell gains a tolerogenic phenotype and secretes the cytokines IL-10, TGF-beta, and retinoic acid. These cytokines induce the naive T cell to differentiate into a regulatory T cell, which will then migrate out of the Peyer's patch and into the lamina propria. Alternatively, the dendritic cell can migrate into a nearby mesenteric lymph node where it can also induce the differentiation of regulatory T cells through the same factors. The regulatory T cells generated then migrate into the lamina propria of the intestine where they secrete cytokines including IL-10 and TGF-beta. The main function of these regulatory T cells is to suppress the activity of effector T cells such as Th1, Th2, Th17, and CD8 T cells in order to create an anti-inflammatory and tolerogenic environment to commensal or food antigens and maintain immune homeostasis. In the next section, we will see how failure in maintaining immune homeostasis can cause disease.
Even though mucosal immunity holds many mechanisms and components in place to maintain homeostasis, there may be times when things go wrong. In some cases, an individual may suffer with inflammatory bowel disease. Mrs. Smith is 65 years old and is complaining about persistent diarrhea, abdominal pain, rectal bleeding, weight loss, and fatigue. You, as a physician, realize that this is a very obvious sign of inflammatory bowel disease. You also realize that you could be dealing with one of either classes of IBD, ulcerative colitis, and Crohn's disease. Ulcerative colitis occurs mainly in the large intestine and colon. The damaged areas will be seen all across the rectum and colon areas, but the inflammation is present in the innermost layer of the colon lining. On the other hand, Crohn's disease is capable of affecting any part of the gastrointestinal tract, all the way from the mouth to the end of the anus. However, it is most likely to affect the small intestine. The damaged areas will appear in patches and tissue and have the potential to damage multiple layers of the gastrointestinal tract. In terms of the mechanism of IBD, we can compare what happens in a healthy tissue versus a damaged tissue. In the former case, intestinal epithelial cells are able to undergo constant shedding and apoptosis. Tissue damage releases danger-associated molecular patterns and starts a protective inflammatory response to restore tissue homeostasis. In the latter case, however, one will encounter non-apoptotic cell death, mucosal oxidative stress, and dysregulation of immune pathways that will release DAMPs at an overwhelming rate. This creates a pro-inflammatory milieu that leads to a heightened inflammatory response through various pathways and excessive intestinal epithelial cell damage. These pathways include activation of innate immune cells, maturation of dendritic cells, pathogenic and APC-related T-cell activation, and chemotaxis of immune cells. You can start off by performing a combination of an endoscopy and colonoscopy in order to acquire imaging of tissue in her gastrointestinal tract for diagnosis. Afterwards, you can prescribe Mrs. Smith medications such as immunomodulators, aminosalicylates, and corticosteroids, which are meant to cut down the inflammation in the GI tract by targeting specific proteins.